If we treat the human body as a state, then cells are its citizens, and citizens must follow rules. Liver cells should stay in the liver. Brain cells stay in the brain. Only in one condition do these cells walk out of their organs of origin, and that is cancer. If I had grown up in the West, I would have pursued a career in molecular biology. But in Pakistan, where I grew up, we didn't have that many options. So I went to medicine as the best thing that I could do to study cancer. But when I saw my first cancer patient, then my intellectual interest became merged with an emotional investment. From that moment on, the rest of my life has to be dedicated to trying to reduce the suffering of these people. I landed in Buffalo, and 5 days later, it was the blizzard of 1977.
Stricken Western New York, an estimated $3 million being.
I was transplanted into an entirely different culture, an alien weather and landscape. I walked into the lobby of Roswell Park Cancer Institute, and you know what I saw inscribed on the wall? If I had a choice between a walk on the moon and saving one life from cancer, I would never look at the moon again. When I read that, I was transfixed by it. I was in a place which can envision a walk on the moon. That's huge. And so, not only does America have the vision, it has the resources to support people to realize their dreams. I was so anxious that there is 6 months before I start my residency. Someone is going to cure cancer in these 6 months. I'll be left behind. But they had a place for a fellowship in pediatric oncology. They gave it to me, and I started working. But within literally a couple of months, it was clear to them that I won't last because I had emotional breakdowns. I couldn't do that to children. So they sent me to the adult oncology. And immediately what became apparent to me in arriving in the adult service was that the very chemotherapy drugs that I was using, those same drugs didn't work at all in the same disease in adults. Even a littler version of our own selves cannot serve as a model for our adult bodies. So how are we depending on animals to serve as model? It is critical to have human samples. And that's when I started banking blood and marrow and saliva and serum, everything I could get from my patients. Today, I have a tissue repository that has over 60,000 human samples stored in freezers. It's the oldest and the largest in the world. We have the perspective going back 35 years on survival. Why did some people with pre-leukemia survive 10 years, and why did some die within 6 months? What were the markers? What was the difference in the two? And this matching of clinical data is so precious. It's an absolute national treasure, and I am proud of it.
Ladies and gentlemen, we are here today for the conquest of cancer, to attempt to find a cure. We would not want to raise false hopes, but we can say this: everything that can be done in this great, powerful, rich country now will be done. And we hope those hopes will not be disappointed.
There was constant hope that cancer cure and a magic bullet is right around the corner. One gene, one abnormal protein, one magic bullet to fix it. We didn't find it. I was sobered into realizing earlier on that in my lifetime, this disease will not be cured by trying to find a treatment for it. It is so malevolent that it's impossible to control it with a few drugs. The outcome for patients today who present with advanced disease is no different than it was in 1930, 100 years ago. Acute myeloid leukemia, same drugs I was using in 1977, I'm using today. Slash, poison, burn. Same old, primitive, hurtful, brutal therapies. What have we really done then? Who's winning the war on cancer? We are spending more and more money, trillions of dollars, on treating cancer patients, and it would be worth it if we were curing them, but we are not. If we want to cure cancer, we have to revolutionize early detection. Stop chasing after the last cell, pay attention to finding the first cell. We can find signals of stress associated with the appearance of the first cell. That's why I started saving cells on pre-leukemia patients, following them till they develop leukemia, saving all the samples, waiting for technology to develop, which is here now. And here is the solution. The cure for cancer is not in this syringe. It doesn't contain any magic bullet. The cure for cancer might be in this needle. What does the needle contain? The needle has a very thin device inside it, developed by biomedical engineers at Columbia University, Professors Sam Sia and Ken Shepard. It can be injected and placed into a blood vessel. It has a computer built into it, so we can communicate with it from outside. It creates an electrical current inside the stent, which is now in the blood vessel. And as cells are passing through, they cause impedance of that electrical field. Depending upon the size of the cell, the impedance signal will be small or large. We suspect that the earliest cancer cell, the first cell stage, produces very large cells. Cancer doesn't arise out of nowhere. There is some stress in the area which is forcing cells to fuse together. The number of genes they need to fight any problems is doubled suddenly. This is the origin story of the first cell. It is a hybrid of two different types of cells that have joined together, like a new species. If, for example, a large cell is passing through that electrical field and gets picked up, we can send it signals to zap it right there. Treat the human body as a machine that can be monitored 24/7. Every biomarker and every cell possible, generating huge amounts of data that is continuously being analyzed by AI for anything going wrong. Then try to reverse those stress markers as they are appearing. This is how we will revolutionize early detection to find a compassionate, patient-centric solution to this problem. I see light at the end of the tunnel. a period of such progress. But it can be accelerated. Instead of taking 10 years, we should take 10 months to do this. It brings me to thinking like Henry Ford, who said, if I had asked my customers what they want, they would have said a faster horseshoe. He created a car instead. These are the problems facing oncologists and researchers. They have to follow what is prevalent instead of doing something radically different. And I was someone who continues to see 30 to 40 cancer patients every week in my clinic. No matter how much research I do, I never lost sight of what is the motivation for me to be a doctor, for God's sake, in the first place. And that motivation is human suffering. And how badly I feel that I have not been able to reduce that at all. I think I don't follow the tradition because I didn't grow up in the tradition. I was an outsider coming in. And so I had the perspective of someone who wasn't raised in this system to follow the beaten old path. No, I was an immigrant, and I had the freedom to really think for myself outside of the box. The lights begin to twinkle on the rocks, the long day wanes, the slow moon rises, the deep moon rounds with many voices. Come, my friends, it's not too late to seek a newer world. That which we are, we are. One equal temper of heroic hearts, made weak by time and fate, but strong in will, to strive, to seek, to find, and not to yield.